Welcome to Biology Access. In today's video, we are going to be doing introductory part of genetics, where some terminologies will be explained. If you are new to this channel, please kindly hit the like button and subscribe so that you get subsequent notification whenever I post a new video. Genetics, as we know it, the father of genetics is Gregor Mendel, and genetics is defined as the study of heredity and variation. Two items or two words are associated with the definition of genetics: heredity and variation. What is heredity? It has to do with the transmission of characters or features or characteristics from parents to offspring, while variation has to do with the differences which we observed. In some cases, they cannot be observed anyway, which is observed or not observed, which can be morphological or physiological, between organisms either of the same species. In some cases, you can decide to look of what different species. In studying genetics, there are various terminologies associated with the studies, and we are going to look at just few of these terminologies. In subsequent video, I'm going to make a full list of explanation of other terms. But the first term here is genotype. Genotype is simply the genetic makeup of an organism. It doesn't change. It's constant. While phenotype? Phenotype is the expressed character, the observable features or expressed character of an organism. In this case, the phenotype can change. The environment can influence the phenotype, while the genotype cannot be influenced by the environment. Let me use a basic example. An organism can have a genotype for being dark in complexion, but as a result of environmental condition or even maybe application of certain cream, it may become light in complexion. That is an environmental influence affecting the phenotype of the organism. The next term is the word gene. Gene, as we know it, is the basic unit of heredity in genetics. And it's actually a section of DNA or a region of DNA that actually expresses a particular feature. Why? Allele is one of the two or more versions of a gene. You can see it here. Allele is one of or two more versions of a gene. For example, let me just give a little explanation of allele. We have a gene for height, and it may have one or two more constructing form, which can be short or tall. So those constructing form of a particular gene is actually regarded as allele. So that's the reason why we refer to it as one or two more versions of a gene. We have homozygote. In term homozygote, it actually means that the two allele, they are identical. While if I use the word heterozygote, remember homo means same, identical, same. Two identical words. The two alleles, they are identical. While in this term, heterozygote, it means two different words, alleles. The alleles are actually not identical, which in this case, we can have a big T representing tongue and a small t. This one is actually heterozygote. While in this case, they are identical. It's homozygote. You can have the small letter T. Both of them are also identical. This is homozygote. So for homozygote, it means the two alleles, they are at a particular gene locus, they are identical. While heterozygote means they are constructing, they are not identical. What is a locus? Though it's not on the board, this is the location of a particular character in a chromosome. Alright? Mendel, the work of Mendel, they are guided by rules. They are guided by rules. The work of Mendel, they are guided by rules. And we call these laws. And we have the first law of Mendel, which is guided as law of segregation. And we have the second law of Mendel, which is guided as the law of independent assortment. What is the first law of Mendel? It states that during gamete formation, each allele, they separate, and they have an equal chance of being passed into a gamete. Let me rephrase it again. During gamete formation, they will separate, they will become single. And each one have an equal chance of being passed into a gamete. So this one can be passed to a gamete. The gamete will be carrying this, or can be passed to a gamete. The gamete will be carrying this. But they have an equal chance of being passed to a particular gamete. That is Mendel what? First law. Why Mendel second law? Is associated with two different traits. For example, we can have a trait for height and we can have the trait for size. Because there are two alphabets, we are talking about what? Two different traits. The way this one is passed to gamete is completely independent of the way this one is what? Passed to what? Gamete. They don't affect each other. Their assortment is completely what? Independent. That is the law of what? Independent what? Assortment. It states that a pair of traits segregates independently of another what? Pair of traits during what? Gamete what? Formation. So this one segregates independently of what? This. That is what? Mendel second law. Now if you carry out a crossing, the progeny or the offspring is called what? The first what? Filial generation. The progeny or the offspring. In layman term, we say the child is called what? The first what? Filial what? Generation. That is the term that is used in Mendelian what? Or in genetics as a course. While the second filial generation is what you guide as what? The grandchildren in a layman word. When you cross this first filial generation or the child, you have what? The second what? Filial generation. In layman word, we call what? The grandchildren. So in subsequent video, I'll be introducing you to crossing, monohybrid, as well as what? Dihybrid cross, and even formation of what? Gamete. Thank you.